Hello and welcome to the Beyond Biotech podcast number 38, and it's going out on St. Patrick's Day. I'm Jim Cornell from Labiotech, and you'll have to take my word for it that I'm wearing green today. I know there are places where they turn the rivers green for St. Patrick's Day or have green beer, and sports teams switch to green uniforms for the day, so I'm doing my bit. This week on the podcast, we're going to talk to some companies that were in attendance at the Hello Tomorrow Global Summit that took place last week in Paris. In spite of the torrential rain and the strikes and the travel woes in the UK because of snow, it was really well attended. And it's definitely an excellent and engaging event. It's not all biotech, but in some ways that's a good thing because it gives you the opportunity to see things outside of the field and see what other great and groundbreaking technology there is out there. And so our guests this week are FreezeM CEO and co-founder Dr. Yuval Gilad, Allogenica CEO and co-founder Inna Menkova, the founder and CEO of MediSieve, George Frodsham, and Poulami Chaudhuri, who is the co-founder and CEO of Helex Bio. And so now it's time for the news you may have missed over at Labiotech.eu. Sanofi added a diabetes drug through acquiring Provention Bio for $2.9 billion. There's a new therapy for the prevention and treatment of COVID-19, and we had an article on how the microbiome can predict if cancer immunotherapy will be successful. Astellas menopause study met its endpoints, but its leukemia drug didn't. Probably the biggest news of the week was Pfizer's $43 billion acquisition of Seagen, and we had some numbers articles, and in ascending order, 5 reasons why collaboration is important in research, 7 biotechs in Hungary, and 10 immunotherapy companies making waves in 2023. Cambrian Bio, which specializes in aging, launched Amplifier Therapeutics, Nanite received funding to deliver genetic therapies to the lungs, and Inspire Biotherapeutics launched to target lung diseases through gene therapy. And of course you can read all of these and a whole lot more at Labiotech.eu. Right, let's get to the interviews. The 8th edition of the Hello Tomorrow Global Summit 2023 welcomed more than 3,000 people to the forefront of deep tech. There were hundreds of meetings, presentations and exhibitors in attendance showcasing science in action, including many biotech and biopharma companies. The grand winner of the Hello Tomorrow Global Challenge 2023 was Sweetch Energy, but a biotech company came in third, Allogenica. Allogenica also won the medical, biotech and pharmaceuticals category, so let's start off with an interview with the company's CEO and founder, Inna Menkova.
So about Allogenica, yeah. So we are a Lyon-based biotech company, preclinical stage biotech company, and at Allogenica we are developing universal CAR-T therapy. Why? Because CAR-T is the revolution for patients with blood cancer. And today, the method by which we are obtaining these treatments is very complex, costly, and sometimes insecure for patients. So because we are basically obtain treatment for each patient from his own cells. So it's called autologous CAR-T cell therapy. And what we try to do is to develop a production method which is universal, meaning that we are not producing from patient cell anymore, but producing from healthy donor cells our treatments. And our technology allows us to get rid of the rejection, which is the main problem when we take cells from one person and want to inject them to someone else. So the idea is we developed, based on our technology, a platform which allows us to produce several candidates. And today we have 3 different programs in preclinical and soon in clinical development for patients with blood cancer, but also with solid cancer.
CAR-T therapy is that it's not really available to quite a lot of people. How do you get around that?
So this is not available to a lot of people because we need to do the production for each person. So today it's 100% personalized medicine. We get rid of it by producing CAR-T, which are universal. We are not taking cells from patients, we are taking cells from healthy donors. And in this way, we can have treatments which are immediately available and to all patients who need it. Because just as a comparison, like autologous method of production allows to produce around 400 doses per year. And with universal method as developed by Allogenica, we can produce 2,000 doses per month. It will be much more easier to administer it to the patient who come to the hospital and need to be treated, because most of the time they are dying, these patients. So basically they don't have time to wait until we produce treatment for them. And also their cells are of bad quality. So sometimes for some patients, we are not even able to produce CAR-T therapies. That's why we are like partisans of universal allogenic CAR-T therapy approach.
Does that bring the cost down as well, the fact that you're able to produce more of them and more quickly?
Sure. The question of rapidity is not really the main one, because since patient is not waiting, we can do our bioprocess in 2 to 3 weeks, but it's manufactured in advance. So for the patient is immediately available. And with the cost, we plan to break it down by 10 as compared to today.
Where are you at with trials?
So today we accomplished the preclinical studies in animal model, in mice model, and we plan first in man by 2026.
You won the biotech sector and came in third overall. Was it a surprise, or did you expect to win?
Actually, it was a complete surprise because, as I told to the whole team of Hello Tomorrow, I was attending actually congresses since I'm student, and biotech companies are never in top 3, so it was really grateful and surprising for the whole team and for me as well.
Did they explain why you were chosen?
We didn't exchange on this point, but some of the person came to me and said that they really liked the application. The first application we did online. That's why we were selected, and they were already pushing for Allogenica candidature to be the winner in the track. And then I think the jury, which was really specialized in cell therapy and CAR-T cell therapy, they appreciated the quality of the project, and also they asked really relevant questions, and I guess I answered honestly, and this was the main key of success in the biotech track first.
Right. Did you have to give a presentation?
Yes, of course, because after we won the track, we needed to pitch in 1 minute on the main stage as well. Basically, I just explained to people how it came into life and into reality for the first patient. And the story was really amazing because like a little girl was dying, and once she had the CAR-T, she's alive today and she has a future and she's cancer free for 10 years. And I just explained that the whole point of Allogenica is to make this happen to all of the patients who need the treatments. And we are not centric on revenues, or we are not centric on someone else. We are centric on patients and their needs.
You mentioned the child that's been cancer free for 10 years. Are you expecting that your technology will be able to get people to a stage where they're completely cancer free, or where is it more managing the disease so that it doesn't progress?
In my opinion, CAR-T is like one-shot treatment, and it is black and white. Or it works or it doesn't work for some reason we ignore. But in 80% of cases, it's worked with 100% efficiency. So you are not managing, you are really curing people from cancer. And in my opinion, this is the first time we can really speak of cure.
And of course, there are lots of different kinds of cancer. Is it better with some than others?
Today, the proof of CAR-T is only for liquid cancer. We still have some problems with solid cancer, and we need to resolve the main problem in solid cancer will be the migration of cells at the site of the cancer. So today we have some tools which can enhance the migration and only efficacy of CAR-T, but so far in clinical trial we don't have any evidences of curing solid cancer because they're also more heterogeneous.
Is that something that you think that CAR-T could be applied towards in the future, or?
Oh, definitely, because I think we have enough knowledge and technology today to design the proper treatment and the proper CAR construct to target solid cancers antigens as well.
So what are the next steps for Allogenica?
Actually, our next step is, in order to progress with our development, we need money. So we are going to conduct the seed round of investment to allow us to produce at industrial scale in bioreactors, cell therapies as developed internally, and then to accomplish all preclinical regulatory studies. The interesting point for us is, like, as compared to other treatments such as bioantibodies, for example, we don't have proper animal model except mice. So that's why we can go quite fast with the development of our technology. And that's why we plan first in man by 2026. So as soon as the investment is done, and I strongly believe it will be the case because people understand and now see the proof of concept of what we are designing. So by this time, I'm pretty confident that it will be okay for us and we can progress with our preclinical development.
I guess winning at the Hello Tomorrow probably helps when it comes to talking to investors, or at least hopefully it will.
Yes, it is, because Hello Tomorrow also is one of our trophies, because we previously won also iLab. This is a French national concourse from the French government. So we were laureate last year in iLab, and also we won some other regional and national context as well.
One startup that caught my eye there was Helex, which is based in India. It's a biotech working on gene editing systems, and rather than me tell you about it and get it wrong, here's the company's CEO, Poulami Chaudhuri, to tell us more. Can you tell me a little bit about the company?
Yes, hi. So we are a precision gene editing therapeutic company. So currently, for the translation of gene editing based therapies, there are two biggest challenges which are hindering the translation. One is the precision and the other is the safety. So when I say precision, when you're doing an within the body therapy, you are targeting the liver, let's say, but the delivery vector actually takes it to lung and spleen as well. So there are chances of editing in those organs as well, which can have serious consequences. And the second is safety. With CRISPR editing or gene editing based therapies, you do not know what kind of an impact is it going to cause or the side effect it's going to cause inside the body. So to understand that, we use a three-dimensional viewpoint at Helix. So we use the 3D vision to introduce precision as well as introduce safety into the gene editing based therapy. So today the current market looks at the drug design for gene editing, looking at the DNA in a very linear fashion, where whether you're making the therapy for liver or lung, the same DNA is considered to build the drugs. But ideally, if you think about it, your eye cells are very different to that of your liver cells and your muscle cells. They all have the same DNA, but it's really the way the DNA is packaged inside them that makes the world of a difference. So we at Helix leverage the 3D organization or nature's own intelligence called epigenetics to introduce that precision as well as to understand the safety. So today we use that to introduce precision into the therapies that we are developing, which are rare disorders, currently focused on the ophthalmological conditions like Stargardt's disease. Tomorrow we are going to partner with bigger biopharma to work on any disease, because the entire platform is agnostic to any disease that we are talking about.
Yeah. And how does it improve on current standards in terms of the speed that you can work at or the accuracy?
Oh, true. So it is built on an intelligent platform. So first of all, it cuts down the amount of trial and error that is involved in gene editing based drug design today. So whatever we learn with every run improves the platform. So to start with, we are working on a particular retinal dystrophy, but as we progress with more and more data, it is going to become a franchisee for the entire ophthalmological condition area. And the same applies to the rest of the body too, because we not only rely on the nature's intelligence, we also leverage the computer's intelligence as well to fast track the entire process. So that is going to accelerate the therapies not just for us, but for any therapeutic company that is developing a gene editing based therapy.
And are you looking for partners or investment? Or?
Yeah, so here I am actually looking for collaborators but also investors. So we just closed our pre-seed round and we're looking to raise our seed round. So identifying strategic investors who would add value to what we're building at Helex, which is to make which is to really make gene editing based therapies a success, and genetic disease is a thing of the past.
What's the reaction been like? Have you had any good discussions?
It's actually been very useful. So I met quite a few good biotech VCs and also met a lot of new startups which are working in the retinal space where I see potential for collaboration. So it's been great.
Next, it's to a biotech company working with the black soldier fly to provide feed for animals. To tell us about it is the CEO of FreezeM, Yuval Gilad.
So FreezeM is a biotech company established in Israel as a spin-off from the Weizmann Institute of Science. Me and my two co-founders, Idan and Yoav, we did a PhD there in different fields in biology. And then towards the end of the PhDs, we came up with the idea of FreezeM, and this is how it was founded. The company is working on the insect protein space, but we're not producing the protein itself. We're enabling others to do it by becoming the seed company of the insect farming industry. We actually do the breeding, the reproduction of the black soldier fly, which is the leading insect in insect protein production. We generate the mating of the flies, optimal mating of the flies, laying of the eggs and the hatching of the small larvae. But then we developed a technology to stop their life cycle after they hatch. So they actually hatch, and before they start their rapid growth, this animal can eat like tons of organic waste and grow very rapidly by 8,000 times in 14 days. They eat waste, convert it into their own body mass, and then you can produce protein and oil out of that to feed animals. And we stop their growth at the beginning, at their initial stage, where they're 0.5 millimeter in size, and that way you can pack them in pre-counted packages, ready to use, and send them to insect farmers that are being established throughout the world. And by that we enable insect agriculture, similar to seed companies in more established fields in agriculture.
What do they do with the product once they get it?
They store it in room temperature, but the larvae don't grow, they stay alive. If, for example, you ship it without our technology, so most of it will die on the way. But we stabilize it and have and enable like a shelf life and a storage of 14 days. And then they just have to open the package, deploy the larvae on organic waste substrate that are used to feed the larvae, and the larvae start growing immediately. And then they grow much faster and complete their growth phase and can be used to feed directly to chickens or being processed into protein fraction, oil fraction that goes into animal feed.
And I guess that you don't have any issues with they don't have to be deep frozen or they don't have to be at a specific temperature.
Yeah, so the name of the company is FreezeM because we started this way. We started with the technology that does deep freezing, cryogenic freezing of the embryo itself, of the egg. And we have this technology, but now we're using it for storing of genetic lines and not for ongoing supply because of the reasons you mentioned. It's very complicated supply, it's too expensive, but for genetic lines it's good because you have no other ways to keep the genetic material alive for a longer period of time. And the generation, the cycle is very short, like 30 to 40 days. So you have genetic drifts in the colonies if you don't maintain this ability. And this was our first technological breakthrough. We managed to do cryopreservation in insect eggs, which was not feasible before. You know how to do it with humans, we know how to do it with bacteria, but in insect there was a technological barrier that we managed to overcome. But take it to the other side for the ongoing supply with the suspended neonates. This is done in room temperature, so you don't have any problem with the logistics of cold chain logistics. You can easily ship it, you can easily use it. We pre-count the packages as well, pre-count the neonates. It's very hard because we we promise our customers you receive a package with 50,000 neonates, but you can't really see what's inside. So we need to make sure that we really pack the number that we say, and you need to make sure that you get what you paid for. So we developed a technology as well to accurately count and dose them into the packaging.
And now it's to George Frodsham, CEO of a company called MediSieve that has developed magnetic blood filtration, a platform therapy that enables the physical removal of specific substances from the bloodstream of patients.
I'm George Frodsham. I'm the founder of MediSieve. MediSieve is a London-based biotech established to develop a technology that enables doctors to physically remove practically anything from blood specifically. So we basically go to doctors and we say, what is there in blood that's causing you a problem? Is there something that's causing a disease or symptom? Is there something that's preventing you from administering the best therapy to this patient? Tell us what it is. We can physically take it out for you and hopefully solve that problem.
And how does without giving away trade secrets, how does the technology work?
It's what's known as an extracorporeal therapy. So extracorporeal means outside of the body. So similar to dialysis, which most people are familiar with, we continuously circulate a patient's blood through this external blood circuit. And within that circuit is where the magic happens. So we actually use nano-engineered magnetic beads that are introduced to the blood while it is circulating outside of the body. And these beads, you can imagine them as little nanobots if you'd like, but they are designed to seek out and bind specifically, so only, to the target that we want to remove. And then we have a filtration system that captures those beads based on their magnetic properties and everything bound to them as well, preventing either the beads or the target from returning to the patient. And that's the method through which we can enable the physical removal of things from blood. And the really cool thing about our tech is that it's easy for us to re-engineer a new version of our beads so that they bind to different things, and hence enabling us to go to doctors and say, what do you want us to take out? We can take it out.
How are you able to distinguish between things that aren't supposed to be taken out of the blood as opposed to things that do need to be taken out?
That's an excellent question. We rely on existing understanding and evidence of disease, and people far smarter than myself who have studied these things in great detail. And what we find is that the role of different molecules or different antibodies or different toxins is often quite well understood, but we simply don't have the technology to enable us to remove those things. And so that's the gap that we're filling. So we are not in the business of identifying new things in blood that are causing a problem. What we do is find things where people already know that these things are causing a problem, and then provide a solution to hopefully just improve patient prognosis, patient outcomes, and often it can be a life-saving intervention as well.
And what kind of things can you remove from blood, and are there things that you can't?
There are a number of programs that we are developing at the moment. Our focus internally is in the gene therapy space. This is what we were talking about when we were at the Hello Tomorrow conference in Paris last week. AAV gene therapies are the latest cutting edge targeted gene therapies developing cures for diseases like Parkinson's, SMA, hemophilia, and a number of other genetic conditions and rare diseases. And they've shown huge promise. They might even be able to develop something for diabetes. And the problem that all of these gene therapies has is that the vector that they use to deliver the gene therapy, so the vector, that means the vehicle, if you like, that's used to deliver them, can be countered by the patient's immune system if the patient has antibodies, pre-existing antibodies, to the vector that these people are using. And the vector in the case that we're talking about is something known as AAV. I won't go into detail of what that is, but What is important is that about half of the population has antibodies to AAV. So what that means is that these pharma companies have developed these amazing new cures for these previously uncurable genetic disorders and conditions, and half of patients simply cannot get them. And that's where MediSieve comes in. So we can physically remove the antibodies from the bloodstreams of these patients. It's a process that takes 2 to 4 hours, something like that, and very low risk intervention. And then that gives a window of opportunity for doctors to administer these ultra cool new gene therapies.
And I see that you have several programs. Could you tell me a little bit about some of those, the things that you're working on?
So that's our current internal focus. We've also got an asset, a product that we've developed that targets and removes IL-6, interleukin-6. So that is what's known as an inflammatory cytokine. It's a signaling molecule that's produced by your immune system in response to an infection to tell the rest of your cells to have an inflammatory response. And the problem, and we saw this in COVID, it's famously a problem in sepsis and cytokine storms, is that the body can overreact to an infection and overproduce pro-inflammatory cytokines, particularly IL-6. And that leads to some very, very serious problems, particularly for patients in critical care units and intensive care units in hospitals, and can lead to organ failure and ultimately death as the body essentially destroys itself. So what we've developed there is what you might think of as a signal blocking tool, is we literally remove the signal from the blood by depleting IL-6. And that's another example of the power of the platform that we've got. You've got these two applications. Number one, where we are enabling gene therapies to be used on up to twice as many people, but we're not directly ourselves treating them. And this other one where we are a novel therapy, a novel therapeutic modality for one of the major clinical problems that affects our healthcare systems and healthcare systems around the world. And then really the sky's the limit. So, you ask, what can we not remove? Well, the list of things we can't remove is smaller than the list of things we can remove. And we've got ambitions in oncology. There's various cancer applications here, which are very exciting. There are applications in autoimmune diseases, in infectious diseases. We have a not-for-profit malaria program. Really, the sky's the limit for this technology. And that's our ambition in the long term, is to provide this to all the many, many, many patients who could ultimately benefit from it. But it's still early days.
You say it's early days. Where are you at with this, and is it available currently?
It's not currently available. It's still under development. We have completed phase 1 clinical studies for the platform itself. So that is the products within our system that enable us to do this across the various disease areas. The phase 1 clinical study looks at safety. So we've performed this on a number of healthy volunteers, 0 adverse events. We're very, very happy with the results there, demonstrating that the procedure is safe. The next step for us is to take one of our 2 assets, either IL-6 or AAV or both, if we get the funding, into clinical studies to prove not only that they work in humans, but that they provide the benefits that we expect them to provide. And that's what you do in phase 2 and phase 3 clinical studies. Then you have regulatory approval, and then it reaches the market. So we're still a couple of years off having this launched in the market.
And what's the reaction been like to it?
Generally speaking, people love it. I think there's an understanding that this is a technology that humanity should have. I like to say that if you saw this thing in like a sci-fi movie, you'd go, of course we'll have that technology in the future. We treat cancer by cutting out tumors. You can go trace back to Roman times, people doing operations to cut tumors out of bodies to treat things. We remove stuff from our bodies as a clinical intervention all the time. Appendicitis and dialysis and these various other therapies. But no one today can remove specific things from blood across a really broad range of different targets. So I mean small molecules and I mean large cells. And if we unlock that potential, suddenly we open a whole new door into opportunities for how to deal with clinical problems that have confounded us for decades in some cases. That's what gets me really excited. And I think people get that and people understand that. So the reaction overall to what we do is really positive. My job as CEO of the company is primarily to make sure we get funding. And conversations with investors, of course, are not just about how wonderful the technology might be, but also how you can ultimately make a return on it. How can this company become profitable? What is the strategy to get through these clinical trials, regulatory approvals, and get to launch? And, those are the conversations that we need to have with the investors and indeed the major multinationals who we want to work with and who hopefully will provide some resources and funding to help us achieve some parts of this dream.
And I guess another aspect of that is probably pricing, because it has to be something that is realistic, especially where only certain people or certain countries have access to it.
Pricing is a major point across the entire world. Look at the NHS, right? Healthcare economics and value for money is a major factor in deciding of whether they're going to offer a treatment or not. And the reality is, even for a life-saving intervention, people put a price on human life. It's different around the world as well. And that's the harsh reality of the world that we live in. There's no point in us developing something that's going to cost, 10 million pounds per intervention. And, it saves one life for every 100 people treated. That's never going to fly, right? So you're right. The cost is a factor. And we've known that from day one, and we built that in from day one, because cost is something that you can incorporate into product design to a degree. So you're right, getting that right in terms of our costs to produce and provide this therapy versus the price it's ultimately sold at. That's an ongoing business challenge that all companies face.
Exactly. And I think that there are other factors as well in terms of if you're able to help people, then it's saving the pressure on the healthcare system, which is a saving of money, which doesn't necessarily factor into the actual cost.
That's exactly right. And that's one of the reasons why the two indications I've been speaking about in gene therapies and in inflammation in critical care units are exciting areas for us, because both of them are potentially cost saving. So on the gene therapy side, if you look at the cost of a gene therapy, they're actually the most expensive drugs in the world, but they are cost saving because the cost of managing these diseases over a patient's lifetime or even just over a few years is so high that a cure becomes very cost effective very quickly. And so if we can enable those cures to reach twice as many people, that we're benefiting the patients and we're benefiting the pharma companies, but we're also benefiting the overall healthcare service. And we are not one of the most expensive drugs in the world. Our intervention will be much more cost effective. And in the other area where we're talking about people who have what are called hyperinflammation crises in critical care units, so this immune overreaction that I was talking about earlier, that causes patients to spend far longer in critical care units than they otherwise would. Every day, I think every 24 hours in an intensive care unit in the UK costs like £2,000 or something in the NHS, right? So if you've got something that means that patients spend 3 days in a critical care unit and survive as opposed to 3 weeks, that makes economic sense. But that's all factored into our strategic thinking and our product development plans as well.
And so where do you go from here?
Well, we're always pushing towards that goal of bringing this to patients. We know the steps we're going to jump through, and ultimately for us, what we need is money. It's as simple as that, because we've got the tech. We've proven that it works. We've got an amazing team. We've got our regulatory documentation in place. We've got our pathway. We've got our plan. We know what we need to do. We know how to do it. We know how long it's going to take. We know how much it's going to cost. So now that's my job, as I said, is I need some people to come in and help us deliver on this.
Was that what you were doing at Hello Tomorrow? Or was it more funding or was it more awareness?
I'm always doing funding. I'm a Biotech CEO. I'm fundraising right now, Jim, in a way.
I guess, yeah.
But Hello Tomorrow, different conferences achieve different goals. Hello Tomorrow for us was more about awareness. It's fantastic for us to be one of the finalists across this incredibly competitive and incredibly broad and well-respected competition. It was an opportunity for us to share with a wider network of investors and people more generally what we're doing, because we're not exactly a household name yet, but MediSieve hopefully will be one day. So it was a lot about awareness, but there were also some great interactions for us with people there, and some potential opportunities as well in that funding department. That's also what that conference was about.
And that's it for another podcast. I'd better get packed for the trip to Basel via Glasgow, Amsterdam and Zurich for Bio Europe Spring next week. Please do get in touch either by email or through the partnering platform if you'd like to meet, although preferably not at 7 a.m. So thanks a lot for joining us, and I hope wherever in the world you are that you have a great week ahead. Take care and join us next week for another Beyond Biotech.